Hello everyone, my name is Katie Kramarczyk and I was the graduate student working alongside Andy Erickson on our biofiltration media optimization project. And so I'm just going to kind of quickly walk through our experimental setup and just some of the things we tested in the last 3 years. So over here you see the tank. This is where we stored and mixed our simulated stormwater. So we just pulled water from the Mississippi River, filled the tank about to 250 gallons, and then added our phosphate, left that overnight. And then what we did was we ran hoses from the tank. We have buckets that hang above for our influent and then buckets that hang below for our effluent. And so we would fill the influent buckets with that spiked river water. We would open all the valves, and then you see all the various mesocosms. Some right now are covered in plastic, and that's because we haven't yet run an experiment this season, and we don't want any rainwater getting in there and influencing, the leaching of phosphate potentially, or plant growth, etc. And so once we begin the experiments, we'll remove those. But essentially, all the mesocosms have a different media blend. Some have biochar, some have water treatment residuals, some are iron and compost mix. So they vary and are randomly placed on the platform. All right, so here's an example of our influent bucket. It's not a perfect example because it doesn't have the tubing attached and the valve, but we do fill it all the way full. Then we'll open that, imagine a valve bottom here, and then we insert the tube into the dissipator in our mesocosm. And you can kind of see that here. So the tube will go in here, and that's how the simulated stormwater enters the mesocosm. The influent is released into these mesocosms and allowed to filter for 2 hours, infiltrate. And then the effluent is collected below in buckets, and then that is where we sample for water quality. So we take samples after 2 hours of letting that filter through the mesocosms, and we then test that water in the wet chemistry lab at SAFL for the concentration of phosphate.